Hi everyone. We left off last time talking about action potentials. So we talked about how this action potential is generated, how it moves along that neuron, and then ends at this axon terminal at the synapse. We took the information from this and created a graph that actually looks like this. So we start at minus 70 millivolts. When that membrane is depolarizing, sodium is rushing into the cell, and it goes all the way up to positive 30 inside the cell. Then we repolarize after we're done firing, and we do that by moving potassium out of the cell. We kind of hyperpolarize a bit right after we fire, and then eventually it settles back at our resting potential. So the question then is, what happens when I get to that synapse? So let's start another drawing. So the first thing we need to do is create our synapse. So we have a neuron that is coming down and it widens into something called the synaptic knob. So my action potential is traveling down this way. And then it's going to connect with another cell, and this might be a neuron or a muscle cell. For now, we'll create this as a neuron. And this connection here between these two neurons is the synapse. So one of my neurons is before the synapse, so this would be my pre-synaptic neuron, and the one after would be my post-synaptic neuron. So what happens is my action potential travels down this neuron, and when this part of the membrane at the synaptic knob becomes depolarized, it causes calcium channels to open. And there's way more calcium ions outside the cell than inside. So when the synaptic knob gets depolarized, those calcium ions are going to diffuse in through calcium channels that open up when they get depolarized. Inside my presynaptic neuron, I'm going to have little bubbles that are storing things. And if you remember when we learned about cells, the little membrane bubbles that store or transport things are called vesicles. So these are called synaptic vesicles. And inside them, we're going to have chemicals called neurotransmitters. Now I'm going to jump to a clear slide here for just a quick second. There are lots and lots of different types of neurotransmitters. There are about 50 different known ones. There are only a few that I'm going to ask you to know, but once you of course get into your program, you're, I'm sure, going to learn about a lot of different kinds. So you may have heard of a couple of these, like dopamine, which from one of the videos you may have learned kind of makes you feel good. And you've got serotonin, which is another kind of relaxed and happy feeling neurotransmitter. So it helps me with relaxing, with sleep, etc. There's a very important neurotransmitter called norepinephrine. Now, epinephrine and norepinephrine are the new names, or relatively new names, for adrenaline and noradrenaline. So when you think adrenaline, you get that rush of adrenaline when you're in fight or flight mode. You're getting that rush of epinephrine. Norepinephrine is a cousin to and very similar in effects to epinephrine. So if you think EpiPen, you're basically giving them an adrenaline pen. So this is going to be an important fight or flight neurotransmitter. So we actually here have a chemical, norepinephrine, that can act as a hormone by being released into the bloodstream and circulating to a cell that has a receptor. Or we can have it act as a neurotransmitter if it's being released at a synapse and signaling another cell. And then one more that I'm going to ask you to know is called acetylcholine, or capital A, capital C, lowercase h. And this is a very important neurotransmitter because it is used by 3/4 of your autonomic nervous system. And we'll talk about that more in another video. And it's the kind of go-to, if you will, neurotransmitter that stimulates skeletal muscle. So right now, while you're taking notes, you're sending signals to the muscles in your forearm so that you can control your hand so that you can write. And every time you're signaling those muscles to contract, you are using acetylcholine, or ACH. So if you were to block all of the receptors for ACH, you would essentially paralyze your muscles. So let's go back here again. We have our synaptic vesicles that are going to be filled with neurotransmitters. Neurotransmitter is a little bit long to write, so let's just pick the most common neurotransmitter you have and put that in here. So that would be ACH. But it could be any of our neurotransmitters depending on where you are in the body. So what happens is the calcium is going to move in through calcium channels that open up when this area gets depolarized. The calcium is then going to essentially knock on the doors of those vesicles, and it's going to trigger them to dump their contents into the synapse. So it basically triggers that. Well, that did not do what I wanted it to do, but we can make that work. Oops. We'll just add our synapse back in here. So what happens is this vesicle fuses with that membrane and dumps its contents out into the synapse. So in this case, I'm dumping ACH. Now with neurotransmitters, they're kind of like letters that one cell is sending to another cell to tell it to do something. If I mail a letter to someone that I know in California, and it travels all the way through the postal service until it gets to California, that's like my action potential traveling down the neuron. But if the person I'm sending it to does not have a mailbox for the letter to go into, they'll never get the message. So in order for this next cell down here to respond, it's going to have to receive this signal. And we do that using protein receptors on the surface of this cell. So the ACH, or whatever neurotransmitter you have, is going to bind to those receptors. And conveniently attached to those receptors are other little proteins that act as protein channels. So we end up with a protein channel here and a protein channel here. And these protein channels open up and allow sodium to go in. And if you remember, back when we talked about action potentials, when sodium flows into the neuron, if enough of it goes in that we hit threshold, then that neuron is going to fire. So when we get to the end of that neuron, I send my neurotransmitters out, they bind to receptors, and sodium starts to flow in. If enough sodium enters that it hits threshold, that minus 55 millivolts, then another action potential is going to be generated. And then everything that we talked about here, so with my action potential and then my repolarizing, is going to happen here in this next neuron. So let's recap that real quick. And don't forget that the steps are laid out for this process in the notes that are posted up on eLearning. So my action potential travels down this presynaptic neuron. It causes calcium channels to open. Calcium flows into that presynaptic neuron, triggering these vesicles to do exocytosis, dumping their neurotransmitters into the synapse. Those neurotransmitters bind to receptors, which open up protein channels and allow some sodium in. If enough of those channels open up and enough sodium enters this next cell, then we generate another action potential. Now, I would like you to follow up this lecture video by jumping out to some other links here on eLearning. So if we go into eLearning under Content, we're going to go into our week 4 for nervous system, and then click on lecture videos. Actually, I want to go under resources, I believe. Yes. So you're going to go under the module, week 4, module 5, nervous system, study resources. And I want you to go to this link called Mouse Party. You're going to need Flash Player. So if you don't have Flash Player, you'll have to download it. Or you can just click on it to allow it to run. And you're going to have a little party. So you're going to have a bunch of mice. Each of them has been consuming or partaking of a particular drug. You can pick which one you want to look at. So this guy's on heroin, for example, and this guy's been drinking too much, and this guy's been doing meth. And you're going to pick up a mouse, and you're going to put him in the chair. And when you do that, he's going to go into this machine, and let me just mute that real quick. It's going to go through the process of what actually happens in the brain to neurotransmitters when you take a particular drug. And what I like about it is it does a really nice job of helping to reinforce these receptors and the synaptic vesicles and the neurotransmitters. So I would recommend doing at least one of those just to get exposed to some of these different neurotransmitters in the brain and what that process looks like. But of course you can feel free to go through as many of them as you want. So it'll walk you through a little step and it'll have you click next, and it'll do that a couple of times, and then it will eject the mouse, and you can pick up another one and look at that if you'd like. So you can find that again under week 4 in your content, and you're going to look under the study resources,